Yeah, hi everyone, and thanks so much, Espa, for having this wonderful event again. It's really always spectacular. I'm going to talk about CD4 T cells in HIV infection. Now, I still don't speak Portuguese, even though it's my 5th year here. So what I decided to do this time, copying Tom Hope, is to use Google Translate to put at least under the headlines. However, I don't know if the translation is right. So I'm just hoping that it sort of conveys the message. If we talk about CD4 T cells, we are actually not talking about a single CD4 T cell, one CD4 T cell, or one subset, or one function, but we have actually this whole spectrum of different CD4 T cell function. So we have like a lot of, a big bowl of different skittles, different function that is contributing to the immune responses. So on the one hand side, we have the Th1 cells that are secreting interferon gamma. That's like the typical CD4 T cells we all have in mind when we talk about CD4 responses. And on the other hand, the Th2 cells, like the old textbooks always talk about the pattern of Th1, Th2 responses. But since then, a lot of CD4 T cells have been actually discovered, like T follicular helper cells, T regulatory cells, Th17 cells, Th9 cells, Th22 cells, and cytolytic CD4 T cells. So we have this huge spectrum of CD4 T cell responses that are all contributing to the responses against pathogens. So from the just to mention basically a few of their function with the T follicular helper cells, we are basically talking about the humoral immune responses. So shaping the B cell responses, having B cell proliferation, memory development, antibody affinity, maturation, and class switching. So the T follicular helper cells, they are very critical for the humoral responses to develop. And in the absence of those responses, we have a very dysfunctional immune system, but also have a lack of good humoral responses. And those humans that have those deficiencies, that have a lack or a genetic defect in this interaction, they are always getting extremely sick. Then we have the Th1 subsets that are not only directly antiviral or antibacterial, but it has been shown that they're also modulating the quality of the CD8 T cell responses. They are important for the maintenance of the effector function and the memory development of CD8 T cells. One important aspect of the Th1 CD4 T cell responses is the macrophage activation, so the phagolysosome activation to kill phagocytized bacteria, pathogens, or intracellular bacteria. Then I'm not going to go through all the subsets, but the Th17 cells important for neutrophil endothelial recruitment and activation. The T regulatory cells that are kind of more like a dampening role of the overshooting immune responses, and the Th2 and Th9 responses that are against parasitic infection, and then cytolytic responses that have been shown to play a role, for example, in EBV infection, or also against cancer and other viral diseases. So why I'm talking about all the different CD4 T cell function? Well, in HIV infection, we get this progressive loss of CD4 T cells. So we have in acute infection already this first, this extreme loss of CD4 memory cells, and then a progressive loss of the CD4 T cells. So we have this spectrum of CD4 T cells, and they are progressively being lost. And so all those functions that I showed earlier are basically impacted by HIV infection. This is basically how it looks like in real data from Merlin Robb in the acute cohort, where we see an acute infection, the loss of CD4 T cells, a slight increase again, and then the progressive loss over time. And this loss of CD4 T cells over time we can actually see those changes in the immune system also in HIV infections. So we have many pathological functions or in immune system dysfunctions that are coming up that are associated with a lack or loss of certain CD4 T cell subsets or associated with the dysfunction of those CD4 T cells. So in the T follicular helper cell compartment, we are seeing the B cell dysfunction, a perturbation of the B cells, a lack of immune globulin class switching. We are seeing for the Th1 subset CD8 T cell exhaustion and a lack of memory development. And also for, it might be that they are playing a role with this exacerbation of tuberculosis in HIV infection because the phagolysosome activation of the tuberculosis bacteria in the macrophages is extremely important for the control of tuberculosis. For the Th17 subsets, it has been implicated for the lack of Th17 cells for immune activation and microbial translocation. T regulatory cells have the lack of T regulatory cells have been implicated in the immune reconstitution syndrome with a disbalance between Th17 cells and T regulatory cells. Parasitic infection associated with the lack of Th9 and Th2 cells. And then cytolytic CD4 T cells that have not been studied that much, but what I mentioned earlier have been shown to play a role in other viral infections like EBV infection. So we have this big glass of different CD4 T cell responses, right? And in HIV infection, we are getting the reduction of the CD4 T cells. So we get less skittles. But what also happens is that not all CD4 T cells are equally lost in HIV infection, but we have some that are basically maintained at higher level and others may be expanding, while some might be completely lost. So the question is, which CD4 T cell subsets are depleted? And I'm very excited to actually hear Danny Duick's talk later, where I hope he's going to talk more about that. But the data that I wanted to share with you is basically how it looks in acute infection just from the memory compartment. So this is basically in the red line, the viral load. And these are the different CD4 T cell memory subsets. And what you can see here is that the effector memory are very rapidly lost over the first 50 days of HIV infection, and then followed by the transitional memory cells that are also decreasing in the first days of HIV infection. In contrast, however, we don't see this pattern for all memory CD4 T cell subsets. We see that some CD4 T cell subsets are actually increasing in the first days of infection, like central memory cells, stem cell-like memory cells, or naive memory cells. Now, this pattern looks a little bit weird, but this is the average of 40 subjects. And what we think that there's a depletion happening at the same time that there is an expansion of those cells. So, and it's actually most of the patients, we see the similar pattern of this double peak. Now, when we talk about CD4 T cell subset loss, I showed you earlier all the different CD4 T cell subsets, and one that has been shown to be extremely lost in acute in HIV infection are the Th17 cells. But also for the T regulatory cells, we are seeing a loss of those T regulatory cells. Now, interestingly, we don't only see a loss of the T regulatory cells in the absolute number, but in the relative number, we see an expansion of Tregs. So we have, on the one hand side, a loss of those cells, but in the whole bowl of CD4 T cells, we're getting more T regulatory cells over time. Likewise, for the T follicular helper cells, we are seeing not a loss of the T follicular helper cells in the chronic phase of the infection, but a relative increase of the T follicular helper cells here in chronic HIV infection on SIV infection, and that is already starting in the acute phase of the infection, the expansion of the T follicular helper cells. So CD4, it is from the CD4 T cell subset that are being depleted. It is also important to know that the antigen-specific CD4 T cells are being affected. And a lot of studies have looked at a lot of different specificities of the CD4 T cells that are being depleted. And basically it goes through all different specificities, like the candida responses that are being depleted. Then here in this study showing, comparing tuberculosis-specific responses in comparison to envelope-specific responses, varicella zoster virus-specific responses, and CMV-specific responses, showing that there's a differential pattern of infection and depletion of those cells. And then here we are seeing that the Ad5 specific responses are also, adenovirus 5 specific responses are also infected and depleted. So HIV is not only going for certain memory subset or certain subsets, but also certain specificities are preferentially depleted, infected and depleted. One subset that is affected by HIV infection is the HIV-specific CD4 T cell responses themselves. So the HIV-specific responses that are recognizing HIV in response, HIV and are expanding towards the infection. And in the study here by Hendrik, you see in 12 individuals that the HIV-specific responses are always more containing HIV, are more infected compared to the general memory population or the CMV-specific CD4 T cell population. Now, but if you're taking a look at the axis, and this is actually also written in the paper like this, that only 0.1 to 5% of the HIV-specific CD4 T cell responses are affected or are infected by HIV. So that also means, when you look at the data in a different way, that 95 to 99.9% of the HIV-specific CD4 T cell responses are not affected by HIV and are not depleted. So you can look at this bowl of CD4 T cells in two different ways. On the one hand side, you're seeing the loss of the CD4 T cell responses, but on the other hand, you can also say, well, you still have a lot of CD4 T cell responses that can contribute to the antiviral immune responses against HIV. So you have, even over the lifetime of the person that is HIV infected, you have strong HIV-specific CD4 T cell responses that can potentially contribute to contain the viral infection. So for us, the question is, how much do HIV-specific CD4 T cell responses actually can contribute to the control of HIV infection? So one of the first studies we did a couple of years ago was to look in acute HIV infection, like that at the very peak of acute infection when the viral load is very high, so at Fiebig 2-3, and ask the question, well, how many CD4 T cell responses are there? What is being recognized by CD4 T cells? And if you look here at the GAG, NF, and envelope, we left the strip in between out, and I show you later why. You see at baseline, at the peak of viremia, already a lot of HIV-specific CD4 T cell responses targeting here P24, P15 region, NF, and the envelope region. What really surprised us with this study, though, when we followed those patients into the chronic phase of the infection, not only acute, but into the chronic phase of the HIV infection, we saw that this pattern of CD4 T cell recognition actually doesn't change over time. What is targeted in high frequency in acute infection is also targeted in high frequency in the chronic phase of infection. And so over time, we don't see a shift in the responses that are being targeted, but also it doesn't look like we see any change in the frequency of the HIV-specific responses. And indeed, when we look at the breadth of that, so how many CD4 T cell responses are being targeted in an individual, we see that over 1 year, there's no significant change in the number of CD4 T cell responses an individual has. Sure, and here in the matched pair analysis, this is not significant from baseline to 12 months, but you can appreciate that there's maybe a slight decrease over time. And I showed you earlier the bowl of the CD4 responses, right? There might be a slow decay and loss of CD4 responses, but it's not that strong that it would be significant in the acute phase of the infection. So you have strong responses even after 12 months of the infection. And it's the same pattern for the magnitude of the HIV-specific CD4 responses. They are also maintained from baseline to the chronic phase of the infection after 1 year. And there's also in the magnitude, so how strong those responses are, there's no change from acute to the chronic phase of the infection that would point to significance. So this was actually the original study, what we did on the HIV-specific CD4 interferon gamma responses in acute, in chronic infection, and this is the reason why we left out the POL region, because there's barely any responses of CD4 T cells that we are detecting. Here in this almost 100 individuals, we see that there's this clustering here in the GAG region, there's NEF responses, and then we have a second cluster here in envelope. So here we look now cross-sectional by the level of control. So how well the patient can control HIV viremia. And what we found was that the people that are very well controlling HIVs, those elite controllers, that they are preferentially targeting epitopes in GAG P24, here shown in dark blue, but are barely having any responses against the envelope region. So there's this little dip in here. There are few responses towards GP120, but the majority responses are all in the GAG region. In contrast, for those that are not controlling viral load, that have very high viremia, we see that they don't have any responses here in the GAG region, but have quite strong responses against envelope. And it looks almost like by the level of control, like a shift from GAG towards envelope, with a better control targeting GAG and no control targeting the envelope region by the HIV-specific CD4 T cells. And indeed, if you're just looking at the ratio of GAG versus envelope, so how much GAG is targeted versus envelope targeting, we see that there's quite a good indicator of the level of control. With those that have high viremia, have a very high envelope to GAG ratio, targeting envelope, not GAG, and the lead controllers have a low envelope to GAG ratio, targeting GAG. Now, I showed you earlier, though, that we see the pattern of targeting not only in the chronic phase of the infection, but we see this at peak viremia. So at peak viremia, we already know which patient is going to make a lot of GAG and which patient has a lot of envelope responses. And so we wondered whether at peak viremia, this can predict for us by the CD4 T cell responses or the strength of the CD4 responses and what protein they are targeting, whether this can predict us what the outcome of the individual is, so if they're controlling HIV better or worse. And so we stratified our patients in envelope, targeting more envelope or targeting more GAG in acute infection at peak viremia. And we see a very clear pattern that the ones that are targeting more envelope have to go early on heart. That was a time when heart initiation was decided by the physician and not was in the guidelines to be started immediately. We see a very clear pattern that those that are seeing more envelope in acute infection are going earlier on heart treatment than those that are targeting more GAG in acute infection. suggesting that the responses of the gag responses in acute infection by the CD4 T cells that are being normally the target of HIV, that they are contributing to the control of HIV viremia. Another interesting observation that we made from the gag-specific responses in elite controllers was that they had more, that they showed the ability to degranulate. Now, what do I mean with degranulation? Well, when a CD8 T cell or an NK cell normally recognizes its antigen and wants to kill the target cell, it's releasing vesicles that are containing perforin or granzyme. And those vesicles are coated with LAMP1 and LAMP2, so CD107A and CD107B. And when those release the perforin and granzyme, those vesicles appear on the cell surface, and we can recognize this act of degranulation by detecting LAMP1 or LAMP2. And we found that the gag-specific responses show more the ability to degranulate upon antigen recognition compared to HIV progressors. So this means theoretically that the HIV-specific gag responses in those individuals have the ability to directly kill infected cells, are being cytotoxic, are being cytolytic. And indeed, when we just look by flow plot, the HIV-specific gag responses of the CD4 T cells contain to a certain extent granzyme B, perforin, and granzyme A. So they are carrying those effector molecules that are associated with cytolytic activity. And in chronic infection, we see about 20 to 30% of the HIV-specific CD4 T cells are containing effector molecules like perforin, granzyme, or A or granzyme B. And we could further confirm this by basically cloning out all our gag-specific CD4 T cell responses. We found that from these clones that we are making, just sorted based on interferon gamma, we found that 20 to 30% of our clones have the ability to lyse in a chromium release assay the peptide-loaded target cells. So also our clones maintain the cytolytic function. Interestingly, we found that more HIV-specific cytolytic CD4 T cells are present in the acute phase of the infection. Over 70% of the HIV-specific CD4 T cells in the acute phase produce cytolytic molecules that are decreasing over time. And we found that the presence of CD4 T cells that are containing one of those effector molecules is associated with a better disease outcome. So we're finding that those individuals that make strong or make cytolytic CD4 T cell responses in acute infection are associated with a slower progression to disease. So we wanted to know in another study whether those CD4 T cells are actually killing virally infected cells. So what we did, we infected CD4 T cells as the target cells with HIV and performed a viral inhibition assay. This is just a comparison to CD8 T cells. CD8 T cells can perfectly inhibit viral replication by 2 logs, but when we sorted on cytolytic CD4 T cells based on surface markers, we found that the addition of cytolytic CD4 T cells is actually also able to inhibit viral replication. Now, in this individual, not as good as the CD8 T cells, but we are seeing also 1 log of reduction of viral load. And actually, when we compared in several patients the ability to inhibit HIV replication by cytolytic CD4 T cells or CD8 T cells, we don't see a significant difference in their ability to inhibit viral replication. So we wondered whether cytolytic CD4 T cells and CD8 T cells are actually cooperating in their ability to inhibit viral replication. So what we did, we performed a simple co-culture assay where we had again our CD4 T cells as target cells infected with HIV, and we had added our CD8 T cells that were inhibiting viral replication. But when we then added also the cytolytic CD4 T cells, we overall get a modest but significant increase in the ability to inhibit viral replication. So now I talked a lot about effector functions of CD4 T cells, like direct antiviral effect of CD4 T cells. I just wanted to mention another very small subset of CD4 T cells that we are detecting in chronic HIV infection in the periphery of chronically HIV-infected cells. Those that work with HIV-specific interferon gamma secreting cells know already how low the level of the HIV-specific interferon gamma secreting cells are that are detected in chronic HIV infection. Now, here we're talking about the subset that are secreting IL-21, are not secreting interferon gamma, are secreting interleukin-21. And in terms of the number of the HIV-specific CD4 T cell responses that we are seeing, as well as the magnitude of those responses, they are much, much lower compared to the interferon gamma secreting CD4 T cells. When we looked at those responses a little bit closer, what we were very surprised about, that they follow a very different pattern of the epitopes that they are targeted. Actually, in one individual only, we found that an IL-21 specific response and an interferon gamma specific response, secreting response against the same epitope. But most of the time, the HIV specific CD4 responses recognizing in the same individuals secreting IL-21 or interferon gamma were recognizing very different epitopes. So I showed you earlier the interferon gamma responses that are predominantly against GAG and a little bit of envelope. We find in the HIV-specific IL-21 responses against the same individuals, we find much more targeting of the envelope region. When we looked at those responses a little bit closer, we see that the ones that are secreting interferon gamma show a typical Th1 or cytolytic CD4 T cell pattern. They express transcriptionally like interferon gamma, perforin, granzyme B, or the transcription factors associated with cytolytic activity like TBAT or EOMES, eomesodermin. And the IL-21 secreting cells are transcriptionally expressing markers that have been implicated to be associated with T follicular helper cells. So here, the IL-21 secreting cells are expressing the transcription factor BCL6, CMAF, but also PD-1, CXCR5, CD14 ligand, or IL-6 receptor. So transcriptionally suggesting that they have a T follicular helper cell phenotype. So we took those cells after sorting in culture. Remember, it's a very small population, so we had to expand them in culture. And also in culture, they showed phenotypically an expression pattern that have been associated with T follicular helper cells. So they are 100% CXCR5 positive, but also high for ICOS expression, low for CCR7, and an intermediate expression of the CXCR3, suggesting that the peripheral IL-21 secreting cells are T follicular helper cells and involved with the B cell responses and are not directly antiviral like the interferon gamma secreting cells. So in the last minutes, I want to talk about another way how CD4 T cells may be modulated in the chronic phase of HIV infection. Now, when the T cell recognizes its antigen from an antigen-presenting cell, it is interacting through the TCR and MHC class II for CD4 T cells, presenting the antigen, or for CD8 T cells, MHC class I. But this is not the only interaction that is actually happening during the antigen recognition, but we have a lot of inhibitory or activating receptors that are involved in the interaction between T cells and antigen-presenting cells, dampening the responses, activating the responses, or even modulating the responses. So there have been many, many different receptors described that are associated or are involved in this interaction between T cells and antigen-presenting cell, like CTLA4, 4-1BB, PD-1, 2B4, OX40, etc., etc. And all those play a role in activation or inhibiting the immune responses that are resulting from this interaction. It has been shown in chronic HIV infection, like spearheaded by the LCMV model, that over time, in the chronic phase of the infection, not only the T cells progressively lose their functions, so losing ability to secrete certain cytokines or perform certain actions, but there's also been described to be an increase over time in the expression of inhibitory receptor. So basically, over time, dampening the responses and probably dampening the damage the T cell responses can do. So we were interested in, this has been described a lot for CD8 T cells, right? The CD8 killer function, asking the question whether CD8 T cells can control or stop to control viral replication through the upregulation of certain inhibitory markers like PD-1 or CTLA4, you all probably have heard a lot about before. Now, we were interested in how actually the inhibitory responses are inhibitory receptors are impacting the HIV-specific CD4 T cell responses. So we looked again in controller and progressor, and first asked the question, well, how are the inhibitory receptors expressed on CD4 cells in comparison to CD8 T cells? And here, progressors and controllers and CD4 and CD8, we looked at 5 different receptors, 2B4, CTLA4, LAG3, PD-1, and TIM3. Now, what you can see, for example, for 2B4 is much higher expressed on the CD8 T cells, both in controller and progressor, compared to CD4 T cells. But in contrast, CTLA4 is almost not expressed on CD8 T cells, but a lot on CD4 T cells. And then we have other ones, like here, LAG3, that is kind of equally expressed on CD4 and CD8 T cells, or PD-1 is also pretty equally expressed on CD4 and CD8 T cells. So these are the bulk cells, these are not the antigen-specific cells yet, right? And what we found is that some of those expression patterns are much more affected by viral load on the bulk cells. Like here, for example, the CD4 cells upregulating CTLA4 in controller compared to progressor, and here also LAG3 being higher in the controller. So this is the overall pattern what we are seeing in HIV infection of CD4 versus CD8 cells. These are not the antigen-specific cells. And what we found is that overall, black means no expression of inhibitory receptor. So we see that much more inhibitory receptors are totally expressed on CD8 T cells compared to CD4 T cells. But we see that in progressors, we see overall an increase in the cytolytic an increase in the inhibitory receptor compared to controller, while the bulk expression of the inhibitory receptors is not significantly different between controller and progressor. What we're also seeing that there's much more double expression of dual expression of two inhibitory receptors, basically making up here PD1 and 2B4 on CD8 T cells, while we for the CD4 T cells see much more single expression, and when we see dual expression, we are seeing the dual expression of CTLA4 and PD1. So our next question was how this is impacting the CD4 T cell response and the CD4 T cell function. At the very beginning, I showed you like this whole picture of the different CD4 T cell functions, and we just looked at a few of them in comparison to CD8, like interferon gamma, CD40 ligand, TNF alpha, and IL-2. And there again, it was interesting to us that interferon gamma in generally was the interferon gamma secreting CD4 T cells were much more impacted or were expressed much higher inhibitory receptor compared to the other CD4 T cell functions. But what was interesting to us that CTLA4 was a little bit of an exception because we found much more expression on the CD40 ligand and TNF alpha expressing cells. I did a mistake here. This is CTLA4 and this is TIM3. So much higher expression on CTLA4 there compared to on the CD40 ligand positive responses and TNF alpha positive responses. Now here also the last two, like LAG3 and 2B4, we found again higher expression on the CD40 ligand and TNF alpha responses, while PD1, for example, what I showed earlier, was higher on the interferon gamma secreting cells. So this is the total picture, what we are seeing. Overall, we find that the interferon gamma secreting cells have the highest expression of inhibitory or modulating receptors, and while we found that the TNF alpha and IL-2 expressing cells have the lowest expressing of inhibitory receptor. Now, with that knowledge, we know that using a blockade, like an inhibitory receptor blockade, that this might impact different CD4 T cell function. It has been shown like a couple of years ago and is used in cancer treatment very successfully, like melanoma treatment, that using a checkpoint blockade, so using an antibody against one of those inhibitory receptor results in better CD8 T cell immunity, and it has been focused on CD8 T cell immunity here. For us, however, was the question, well, how does the checkpoint blockade actually impact the functions of the CD4 T cells? And this is still preliminary data, what I'm showing from now on, but what we found was that in generally, the interferon gamma and that the PD1 and CTLA4 blockade had the strongest impact on the CD4 T cell function and was following a very similar pattern to the CD8 T cells. Now, we found a significant increase in the interferon gamma and responses for blockade with PD1 and CTLA4. Same for CD40 ligand, but there also 2B4 had an impact on both CD8 and CD4. TNF alpha was only impacted by PD1 and CTLA4 blockade. And lastly, IL-2 was only the IL-2 secretion. We found only significant impact after PD-1 blockade in the CD8 T cells. So this means also in a different, when you look at this data differently, that the 2B4 blockade is the most targeted impact on the function, while PD-1 is the most broad blockade, has the most broad impact on CD4 and CD8 T cell function. So we wanted to test this in an animal model, what the different blockade actually, how the different blockade impacts the CD4 T cell responses and CD8 T cell responses, but also wanted to understand whether this has an impact on the disease outcome. So at our university, there's a friend virus model very established, what I didn't really know much about before as well, but it's basically a model that has been used for HIV, to study as a model as HIV infection. So as you will, it's kind of a model like the LCMV model, but not as much used as LCMV. The friend virus is a gamma retrovirus. It also causes persistence in the target cells, which is here erythroblasts, myeloid B cells, and to a lesser extent also in T cells, while you see in HIV CD4 T cells and plus minus macrophages. And here the compartments that are impacted are in the friend virus, the bone marrow and the lymphoid organs, while in HIV we predominantly see effect in the lymphoid organs and ZNS. We have also, like in HIV of course, to the loss of CD4 T cells, we get the typical AIDS picture, but also in the friend virus model, we see cancer development in the mice as erythroleukemia. So they are more developing a direct cancer caused by the friend virus. So we use this model to basically understand whether the blockade of the different receptors has an impact on the outcome of on the viral load in those mice. So what we did, we used the same model of mice and that are infected with friend virus and added either anti-CTLA4, anti-LAG3, anti-PD-L1, or anti-TIM3, and then asked the question, how is this impacting the CD4 and CD8 T cell responses? Overall, we see that just if you purely look at the expression of the markers in the mouse, we see a little bit different pattern in the mouse compared to the humans. We see much higher CTLA4 expression also on CD8 T cells, but the highest expression of LAG3, and after infection actually rather low expression in comparison to the other ones of PD-L1. So it's not directly translatable to the HIV human model. However, what was interesting to us was that a CTLA4 blockade was the only one in that model, even though we had a high expression of LAG3 and TIM3, we did not see any significant impact on the blockade of those receptors, but the highest impact on the viral load of those animals, or the number of infected cells, was through the blockade of CTLA4. So we're now continuing trying to understand whether this is an impact on the CD4 or CD8 T cell responses, but unfortunately we don't have the data on this yet. So to sum up what I showed you in this overview on the CD4 T cell responses, is that CD4 T cells are differently impacted by HIV. We are detecting in almost all of our individuals CD4 T cell responses, and but what is interesting is that some of those HIV-specific responses are not only interferon gamma secreting, but have a direct antiviral effect. And that why we see a loss over time of the HIV-specific CD4 T cell responses because they're infected and depleted, But this is not as strong over the time of 1 year into the chronic phase, so that even in the late chronic phase, in the high varying progressors, we can still detect antiviral HIV-specific CD4 T cell responses. I've shown you that about 20 to 30% are directly cytolytic. They can inhibit viral replication, they can cooperate with CD8 T cells, they have a very distinct profile of expression of perforin, granzymes, and they are actually also transcriptionally different, what I didn't show you here, compared to the Th1 cells. And I've shown you that we have, compared to CD4, CD8, a very different profile of inhibitory receptors on the CD4 T cells that also can modulate the function of the CD4 cells against that are targeting HIV. We find the highest expression of CD84 on Th1 CD4 T cells, and at least in the mouse model, this has the strongest effect on viral load, but there also CD8 T cells express higher levels of CD84 compared to the chronic HIV infection or CD8 T cells in HIV infection. Well, thank you again so much for this course, Esper. Thank you for listening, and I take any questions.